You can front it. Right. Good evening, people. Good evening. We will discuss paper number 5. Can you hear me clearly? Balan lamai. Hello. Can you hear me clearly? Yes or no? Good evening, Danisha. Yes, clear. Ne? Right. Paper number 5, people. Did you answer paper? Did you upload the paper? Yes or no? I think I gave you the paper last Saturday. When did I give you the paper? Last Saturday, ne? Last Saturday? Is it a week before? Last Saturday or week before? Paper number 5. Hello? Last before, ne? Last before. Okay. I think then we had to give paper number 6 this Saturday, ne? Along with the paper 5 market scheme. I had to give you on this coming Saturday. So we'll do that. People will quickly discuss the paper. So please upload your papers. Especially people, we can correct your essay part, right? So there are so many weaknesses, people, in you. So we can fix them. We have time. And when you answer the paper and upload it, people will mark them, hand over to you. We'll go as a routine, 2 papers per month. So we can go. Now, one of the latest finding says that people, the best way to prepare for an exam is the do practice questions, right? Best way to prepare for an exam, right? To ready for an exam. You are getting ready for an exam. So therefore, people, compared to the studying and preparing short notes, and then people, the various other activities, reading, highlighting, right? The most effective way of learning is the practice questions, right? So this is the golden hour, right, of the week. Golden hour of the week. Golden 1.5 hours, we can say, right? Any student who are expecting to go to medical faculty, right, and for the other faculties, people should not miss this time, right, part of the week. And the best thing is this is an open class, right? You can join, anyone can join at this time period, right? Golden, right? Very difficult work we manage, and then people make the things available for you. So it's good that a few people take the benefit of it. Otherwise, you get more competition, right? So it's better, right? From your point of view, people, it's better, right? It's better from your point of view. You have less competitors. So don't get discouraged when few people are joining with the class. You have to be so happy about it. Right? You have to be so happy about it, and then join, right? Because your courage and then this working will take you to the right to your success. So let's start the day with that. So people, the questions. So give us some momentum to study hard. Yeah, we'll talk about that further. Okay. This is enough. You have already your working people. Maintain it, right? Do more questions, right? So you will find, you will reach to your goals at the end, right? Continuous working is the best thing, right? Don't give up. Difficulties come. No one can do this, people, without any problem. Comfortably, with full comfort, people, you can't pass an exam, right? You have to go through the challenges. So a lot of people are having a lot of issues, but that's how life goes. The people who have won their life, who have realized the dreams, have not done it, people, without any troubles. All have done with challenges and all. So work with challenges, right? People, we are going for the paper. We'll start with the discussion, people, of the essay paper. starting the discussion with the essay paper. I'll give you a printed version of this marking scheme. Describe the nuclear division process that forms two identical nuclei. Nuclear division process that forms two identical nuclei. Now, you know, nuclear division, two identical nuclei, the nuclear division process of making two identical nuclei, it's mitosis. And then when you are describing this mitosis, people, you have to describe only the nuclear division steps. So talking about the interface is not going to be helpful. Start with the prophase, pro-metaphase, metaphase, anaphase, and telophase. That's the question, right? That is the question. The boundary limits, right? The answer limits are there, and you can right? You can write within that frame, right? So your answers, whatever you submit now for us, we'll check that, mark it, and then give it back to you. People, these are the basic points, right? These are the basic points. If you have not written the answer yet, I think you can complete it and then submit it, people, within as soon as possible, right? Maybe today or tomorrow. So we'll mark and then give it back to you. The nuclear division get divided into 5 stages. The prophase is there. Chromatin fibers get condensed by shortening and thickening, right? And then they transform into chromosomes. And then as a result, chromosomes become visible under the light microscope. Nuclei get disappeared and the chromosome appear with cystochromatin. That happened during that. Nucleolus disappear. Nucleoli disappear. And then chromosomal arms, cystochromatin is attached at the special portion, cohesin attach them. And then people, the formation of mitosis spindle begins. And then we come to the next phase. What is that, people? The prometaphase, right? And prometaphase, nuclear envelope fragments and chromosomes are going to release to the cytoplasm. And then, you know, the spindle fibers attach. Kinetochore fibers attach. Spindle fibers go and attach to the kinetochore, moving chromosomes forward and back, and then begin to align them, people. And then comes to the metaphase. The centrosomes reach to the opposite poles, and then chromosomes are going to align at the metaphase plate. And at the end, we can see that the the kinetochore attachment is going to be completed. Right, chromosomes are aligned at the middle equatorial plane. Those points you're right there, people. And then the anaphase, what you have to say that the centrosome, centromeres split, and then sister chromatin are going to separate at cohesin, and they move to the opposite poles. Sister chromatin are separating. As they separate, you know, people, they transform it back into chromosome. We discussed these things last week, right? Supporting you to answer this question. And then anaphase goes into the telophase. So those are the details. Telophase, people, nuclear envelope reforms around each set of chromosome at opposite pole, nuclei disappear, spindle microtubules get depolymerized, right? So those details you write. You are submitting your paper, so we'll mark and then give you right marks for that. The printed marking scheme, people, I'll give you on Saturday, because your suggestion, you suggest people not to give the marking scheme earlier, so I'll submit this on coming Saturday. Okay, but if it is not set with you, let me know. I can adjust anything, people, right? Then people, second question, what is an enzyme? And then part B, list out characteristics of enzymes. And then people briefly describe the factors affecting the rate of enzyme reaction. Briefly describe the characteristics affect the rate of enzyme reaction. Sorry, briefly describe the factors affecting the rate of enzyme reaction. Factors affecting, you know, different factors are affecting. So briefly describe. So what is an enzyme, people? They are expecting what? The question word is what? So there must be a straightforward answer. You must write the definition here, right? What enzyme is, right? The enzyme is a macromolecule which acts as a biological catalyst, and which is produced inside the living cell. That sort of an answer, the macromolecule which is a biological catalyst, right? Some of you give the definition here. Second part, list out characteristics of enzyme. People, we learn how many characters? Around 12 characters we learn, and then you have to write them as a list. You remember those 10. The Nisha says 10 characters. එතකොට 10 characters, most enzymes are globular proteins. We can say enzymes are biological catalysts. And then most enzymes are heat sensitive. And then their presence does not alter the nature of the end products. Enzymes are highly specific to substrate. See, they have written these things, ne? And then most of the enzyme reactions are reversible. And then people rate of enzyme activity is affected by pH, temperature, substrate concentration and all. And then they are being used up during the reaction. They are not being used up during the reaction. And then enzymes has an active site where the substrate come and bind. And then some enzymes need non-proteinaceous component for their action. What do you call them, people? Enzyme cofactors. Cofactors are there. So those are the points, right? I'll give you the point breakdown. Right? I have taken around 14 points here. So 10 points, but 10 facts, but 14 points there. Okay. So that is what you have to write there. Briefly describe the factors affecting rate of enzyme action. Factors affecting rate of enzyme action, temperature, pH, substrate concentration. What's the last one? What's the last one? What is it? Inhibitors. Inhibitors, reversible, irreversible, competitive, non-competitive. You must write them. So here people, as the marking scheme, briefly describe the factors. pH affects the rate of enzyme activity, different enzyme function at different optimum pH values. There is a particular pH range that the enzymes are working. And within that, there is a particular pH value enzymes show their best activity, optimum pH values. Temperature effects for the enzyme activity as well. And then people at low temperature, due to lack of activation energy, there is no collision between the enzyme and the substrate. So no reaction at low temperature. As you go on increasing, the probability of the number of collisions are increasing between the enzyme and the substrate, increasing people the product formation. Really unknown. And then people at optimum pH around 35 degrees Celsius. 35, what we said, optimum pH temperature 35. 35, the enzyme shows their maximum activity. Above that, the enzymes get denatured due to alteration of some of the hydrogen bonds and other weak interactions, and as a result, the denaturation occur. As a result, enzyme reactivity comes down. Those details you must write there, people. What's next, people? Substrate concentration, you write there. The substrate concentration, as you increase the substrate concentration, enzyme reactivity increases, and at a particular point, the enzyme becomes saturated with the substrate, and then people reaction reaches to a plateau. So those details. And then people inhibitors, reversible, irreversible inhibitors are there. Irreversible inhibitors, toxins and poison, we said. It was say reversible inhibitors we can classify as the competitive and non-competitive, right? Their effect, people, you can briefly describe. Right? Their effect you can briefly describe. Their effect you can briefly describe. Competitive inhibitors. They are structurally similar to the substrate. So therefore, they compete with the enzyme for binding with the active site. And then the competition. And as a result, when the inhibitor is available, chance of the substrate interacting with the enzyme decreases. As a result, reactivity comes down. But if you can overcome this by increasing substrate concentration, that is what we call as the competitive inhibitor. You can overcome the inhibition by increasing substrate concentration. How is it? You say non-competitive, not structurally similar, does not bind with the active site, bind with some other site. What do you call that other site? What do you call this other site? What do you call that other site, people? Allosteric site, but what we said, inhibitor binding site. And then the enzyme active site shape is going to alter, reactivity decreases, and then finally what you see, people, reaction comes down. Can you overcome this by increasing substrate concentration, people? Non-competitive inhibition, you cannot change. So those are things that you learn. So this is really productive. Productive, I can see that students are like, right, sending me the answers and all. So I'm so happy. I'm tired, of course, right? I had my morning class in Kurunagala at 8 a.m. I got up at 4 a.m. in the morning. Then I left home people 6:30, reached there 8 o'clock, 8 to 5:15 the class, 5:15 to I came here people around 7 o'clock drive. Now after 1 hour break, people, I'm with you, right? Till another 1 hour, 9:15, right? So you all really give me energy. These people who are chatting with me right now, right? That's good, right? We all have to work together, to to improve, right? You know that the story, there was one flock of birds, captured by a hunter with a big net, so they all worked together, fly with the net, right? For to find their freedom, same like that. We have to work together, as a team. Everybody, you and me, right? Luxury uncle, we all have to work together so that we can improve, all of us, and you can achieve your life goals as well, so that one day you can go for your dreams. When you hit your goals, A level goal you do, and your higher education goal you do well, and your job goal you can achieve, and then finally people, you achieve your dreams, finally you go to your dream. So that's how it should be. And then people moving to next one. Moving to next one. Write short notes on phospholipids, cilia flagella, linear electron flow. That linear electron flow from photosystems. Did we talk about photosystems? Did we discuss photosynthesis? We discussed, didn't we? Yes, we did. So linear electron flow. So phospholipids. What are you going to say about phospholipids, people? They are the lipids containing phosphate. They are membrane constituents. Phosphate gives the negative charge. Phospholipids main constituent on the cell membrane. What else, people? Additional polar molecule is there. What do you call that? Choline. What do you call that? Additional polar part is choline. So these are the points, people. Phospholipids are major component of the cell membrane, composed of 2 fatty acids, 1 phosphate. Phosphate group is giving negative charge. Typically, additional polar molecule choline is there. 2 ends of the phospholipids are different in behavior. Fatty acid tails are there, non-polar. And then people polar head is there. So that's the one of you say that one day when I come to higher position in my path of science, I will definitely mention you as my biggest motivation. And thank you, that's a motivation for me also. That's why we said we had to work together. And then people, cilia flagella. Cilia flagella. Definitely you can go for a better goal. We always try to lift you up. We don't want to see you at our position. We want every time to see you people at a higher level than what we do now. So I'm with you, and always my blessings are with you. So work hard. We can do this, right? No doubt about it. Right? You work hard, right? I am working hard. You work hard. Then it's going to be a simple goal. Right? Never going to fail. Don't worry. And then people, cilia flagella. Cilia flagella are membrane bounded structures, 9 plus 2 structure percent, attached to the cell body with the basal body. And ciliated flagella containing ones are flagellated. What else we said, people? Structure, 9 pairs of microtubules present in a cylinder with 2 single microtubules at the middle. 2 single microtubules at the middle. That's the arrangement. Anything else, people, you would like to mention about cilia flagella? Let's see, people, the marking scheme. Cilia and flagella share some common structures. Flagella are long, elongated structures, and cilia are short structures. Cilia more numerous. I forgot that name. Cilia more numerous. Flagella less number is there. and bound to a basal body, 9+0 arrangement of microtubules in the basal body, and then locomotor appendage, and then move the they are present in the oviducts. Was it there? Oviducts, and they are present in the respiratory passage, the right, and yes. Then people linear electron flow, linear electron flow, linear electron flow, linear electron flow. Yeah, Selvanathan says some information about the linear electron flow. Linear electron flow, people, occurs between PS2 and PS1. The electrons released from hydrolysis of water goes to the how do you describe that, ne? I think that's not a good point to start, ne? How do you describe people this electron flow? We'll start with the photosystem. I'll leave my paper down and then I'll explain like this. Photosystem 2 receives light. Photosystem 1 and 2 both receives light, and then these light energy received by the center complex pigments, they get excited and then emit electrons. These electrons flow along the electron carrier. They are accepted by the primary electron acceptor, pass along the electron transport chain, and then electrons emitted from PS1 goes into NADP reductase and then finally form NADPH. And then electrons released from PS2 reaches to PS1 and neutralizes the oxidized pigments and help them to reduce. And then the electrons, hydrolysis of water is releasing electrons, which is involving in neutralizing the oxidized pigment present in PS2. And then people, the process which happen in PS2 is emitting molecular oxygen. As electrons are flowing from PS2 to PS1, the ATP production take place, which we call as a photophosphorylation. And that's it, ne? Those are details, ne? Right? So Santosh say key of this energy transformation is occur as electron flow take place in one direction. PS2 excite by getting light through excited electrons flow, electron acceptor, hydrolysis occur and neutralize P. Yeah, that's good, Santosh. Right? You are in the track. So look at these people. Linear electron flow. The striking of photon of light on the pigments result in excitation of electrons from photosystem 2 to a high energy state. These electrons will accepted by the primary electron acceptor of photosystem 2. Splitting of water take place as a result, release oxygen, H plus ions and electrons. Electron release as a result of hydrolysis may neutralize photosystem 2. It goes like that. It was say striking a photon of light on photosystem 1 excites P700 to high energy state. Excited electrons will accepted by the primary electron acceptor of PS1. Excited electrons of PS2 at primary electron acceptor will pass through an electron transport chain and reaches to the PS1. The energy released due to the passing of electrons from high energy state to the low energy state is involving in making ATP. Now you can remember that part. Ne? When you do 2-3 times, well, then you remember how it works. The key points, ne? Electrons flowing from PS2 to PS1 is involving in formation of ATP. This is known as photophosphorylation. Excited electrons of PS1 at primary electron acceptor PS1 will pass through electron transport chain and reduce NADP plus and yield NADPH. So these things, people, in any order you can write. Any order, right? In a clear way, you can write that. So marks are allocated for that. So I'll give you the marking scheme. I have the scheme with me, right? I'll give you the marking scheme on Saturday. Right. Now, shall we move people for the structured part? Structured part. It's real difficult. it's real difficult when we discuss without the marking scheme on hand. I find it's difficult. I feel like now it's better people you all have the market scheme with you and then we discuss. I think it's like a little easier method, little easier method. Anyway, feel like it's like more convenient method. Let me see, people. Let me see whether I can show you the market scheme open. Just give me 1 minute. Just give me 1 minute. Right. Check this market scheme, people. Right. First question. State 3 differences between light microscope and electron microscope. Right. Light microscope and electron microscope. Glass lenses are used in light microscope. Powerful magnets are used in electron microscope. That makes a difference. And then image is directly detected by light microscope, but micrographs are used. What are micrographs? Will photos neither. Micrographs are used. Micrographs are used in electron microscope. Both living or non-living can be observed in the light microscope, but only non-living objects can be observed under the electron microscope. Actual colors can be observed in light microscope. Every time people, black and white images can be observed. Dyes are used to stain the specimen in light microscope, but we are using heavy metals to stain in electron microscope. We say stains, neither. Dye is a coloring material. Stain, heavy metal stains are there. Okay. So 3 marks, people, for writing any 3.3 marks. And then people, then what is the level of organization of matter that shows all characteristics of life? What is the level of organization of matter that shows all characteristics of life? We say that's the cell. Cell is the level. So 1 mark for that. 1 mark for that. Some common functions of SCR and RER. State a common function of SCR and RER. Produce transport vesicles we can take as a common event. Both are involving in forming transport vesicles. And then people, there is a diagram which is showing you stage A and B. Stage A and B. What is the stage A, people? What is stage A? Prophase. Stage A is prophase. So how do I say that it's prophase, people? Because you don't see the nucleolus. Nucleolus is absent. Nuclear envelope is still there. So it's prophase. Mitosis or meiosis? Definitely this is mitosis. Right? And then what is the stage B, people? That is the metaphase. That is metaphase of mitosis. So nuclear envelope present, nuclear envelope not present. Chromosomes are not attached to kinetochore. Chromosomes are attached to kinetochore. Chromosomes are not attached to spindle fibers. Chromosomes are attached to spindle fibers. So I'm giving you 6 marks. Giving you 6 marks for writing that. State difference of a tumor and a cancer. State difference of a tumor and a cancer. What are you going to say, people? A tumor is abnormal cell remain at original site. Cancer cells are invasive and attack one or more body organs. State 3 ways of formation of ATP in a cell. There are 3 ways, neither. What are those 3 ways, people? Photophosphorylation, substrate phosphorylation or substrate level, both correct. I think in the resource book, they have mentioned substrate phosphorylation. So we'll follow that. And oxidative phosphorylation. What is activation energy? Minimum energy needed to start a reaction. People, I think that answer is sufficient. But there is no definition given for activation energy in the resource book, right? So we'll take it like that. I think one of you asked that question in the class last Saturday. What is the definition of activation energy, right? Minimum energy needed to start a reaction. I think that is a sufficient answer. This graph is shows you relationship between activation energy and enzyme. Identify A, B, C. What is A, people? A you can take as the activation energy of activation with the enzyme. Right? What is B, people? Energy activation without enzyme. Then what is C, people? C is free energy. Can somebody tell me what we call it, people? Actual, what do you call that? Gibbs free energy. Gibbs free energy. Then people, what are cofactors? What we said cofactors. Cofactors are non-proteinous components which are essential for the catalytic activity of certain enzymes. Non-proteinous components. So 2 marks goes for that. Lavai marks balanda, right? Marks balanda. This one scores 3 marks. State how cofactors different from enzyme activators. How cofactors are different from enzyme activators. Now we say, people, enzyme activator, cofactor and activator. Now, now we will cofactor and activator. This is also not present in our resource book, right? Cofactor is necessary. Enzyme cofactors are necessary for the action of some enzymes. That is what we said. Some enzymes and their function, cofactors are necessary. Activators are not like that. They are not necessary, but they only speed up the chemical reaction. Sorry, they only speed up the enzymatic reaction, right? So the activator speed it up. Activator speed it up, right? Some cofactors different from enzyme activators. We learned that. So 2 marks for that, people. Anyway, this statement is not in the resource book. Part C, 1, define aerobic respiration. Define aerobic respiration. Aerobic respiration, process of synthesizing ATP from the respiratory substrates such as glucose in presence of molecular oxygen. This is called as the aerobic respiration. And then complete the following equation, people. This one is pyruvate. 2-pyruvate, 2 coenzyme A, NAD plus forms 2-acetyl coenzyme A, carbon dioxide and NADH. So to fill in the blanks, I'm giving you, people, 2 marks for that. State the site of tricarboxylic acid cycle. State the site of the tricarboxylic acid cycle. Tricarboxylic acid cycle, we have another name. What do you call that, people? Citric acid cycle. Tricarboxylic acid cycle, citric acid cycle, Krebs cycle, all same, right? So mitochondrial matrix. What is the process of forming ATP in TCA cycle? That's a good question. That is a good question. What is a method that ATP production, people? Substrate phosphorylation. 1 mark for that. State the number of reduced FAD, that is FADH2, and reduce NAD, NADH2 formed in Krebs cycle. NADH, people, 3 produced per 1 acetyl group. 3 produced for 1 acetyl. Okay, that's not wrong. Yeah, 1 acetyl, 3 NADH. Or else, can you say it for pyruvate? I think acetyl better, ne? Acetyl better, right? We are talking only about the Krebs cycle, right? Krebs cycle. So per acetyl, we'll talk about it. Did we discuss people's cellular respiration? Yes, neither. We discussed cellular respiration also, ne? Did we discuss cellular respiration? 16 new messages. Hari. Did we discuss cellular respiration? Yes, ne? We did. Ne? What else you find difficult, people, to discuss? What else you find difficult? You said cell cycle, I discussed it. Cellular respiration, photosynthesis, we discussed that. Anything else that you find in unit 2 difficult? Anything else you find in unit 2 difficult? Or else we can go for unit 3. Unit 3. If you ask enzymes, look. Enzymes, you want me to go through enzymes? Right? If I ask you what difficult section that you have in unit 3, what are you going to tell? What difficult part you have in unit 3? Sure. Sure. What the difficult part? I'm asking unit 3, man. Not in unit 2. Unit 3, what are you going to say as the most difficult part in unit 3 is? I know that you hesitate to say. Namidu said that. It was a Santosh also said that. So, Plante, expecting that. Plante. Did we finish Plante? Still we are discussing Plante. No? Just finished. Ne? It's like just married. Just married. You find that you find that trouble, ne? Right? It's like just married. Ne? Even the honeymoon is not over with the Plante lesson, right? Still you are in the honeymoon. Why you say that? It's, yes, sir, this Plante, that's my mistake. Mistakenly, you went through that lesson, Plante. Yeah, we finished it. So just go through that. Find those plants outside environment. Go and find them. You will find that lesson interesting, right? Okay. So that's a nightmare for me. Discussing Plante in a 1 hour class, in night class, people like this. Oh my God. Right? Fungi is better. Right? The Plante is worst. Anyway, we'll do, we'll try to, but I need some more time. We'll go through enzymes. The next week we'll go through enzymes. If you want, we go through introduction to biology also, and cell structure and function. But anyway, people, it's very short, within a very short time period, we revise people's cell cycle, enzyme, photosynthesis, cell respiration. Those are the parts actually you will learn for a quite long time. Right? All right, we are in Animalia now. Right, moving to next page, people. Fill in blanks to show how many ATP are produced during aerobic respiration. As ATP in glycolysis 2 ATP, that's like net total is going to be 4, net 2. From NADH 5 ATP, 2 NADH produce, each NADH produce 2.5. From 2 NADH, it's 5 ATP. Pyruvate oxidation from NADH 5 ATP. Pyruvate oxidation to NADH is produced, so 5 ATP for that. And citric acid cycle, as ATP, 2 ATP produced from NADH. How many NADH are produced in citric acid cycle? Citric acid cycle, how many? NADH, 6, neither. 6 NADH, is it 6? If you take citric acid cycle, only 6, neither. 6 into 2.5, that is people 15 ATP, and FADH2, 2 FADH2 produce, each 1.5. With 2 people, then it comes to 3, right? So that's the total number, right? And then people will come to the next question number 2. State sequence of 4 stages to form the first cell in biochemical evolution: abiotic synthesis of small organic molecules, polymerization of these small molecules to form organic macromolecules. Organic macromolecules are packed into membrane to form protocells. Nucleic acid gained self-replication capability, which made inheritance possible, right? And state main differences between artificial and natural classification. Classification done based on evolutionary relationship is called as natural classification. And in artificial classification, evolutionary relationships are ignored. What is the reason for Haeckel to introduce kingdom protista? With the discovery of the microscope, they found that there are some organisms which cannot be included as plants or animals. To get over this difficulty, he introduced the kingdom protista. 2 marks for that. State biological definition of a species. Biological definition of a species: group of organisms who are sharing similar characters and has the ability to interbreed and produce viable and fertile offsprings. We call that as the species. That's the biological definition. We learn, people, there are so many species concepts are there, because sometimes we can't depend on a biological definition. You learn them. State 5 codes of binomial nomenclature. 5 codes of binomial nomenclature. 2 species of organisms can't have the same name. Each species have generic name and a specific epithet. Together make the species name. Name should be made up of Latinized words and written in Roman scripts. Name should be underlined when handwritten and should be italicized when printed. And then the first letter of the generic name should be capitalized and the rest of the letters must be in simple letters, right? And then you can say further, the name of the author is abbreviated at the end with a single letter or with an abbreviation. And what else we said? Third word can be introduced to denote the subspecies and all, right? So rules we learn. This is the nice part. This is nice part, right? What is that? The dichotomous key. Dichotomous key. Is there anyone find people dichotomous key difficult? Right? Is anyone to find dichotomous key difficult? We discussed it recently. So you have to find, people, these 4 organisms. I gave you the same question, people, in the I think I gave you a similar question or the same question, people, in the book, your practice questions. So here the insect has 2 wings. Who are the insects with 2 wings, people? This guy has 2 wings. This guy has 2 wings. And that's it. Those 2. So the insect with 2 wings. Insect has more than 2 wings. The rest of the ones. So A, we select the organism A. Insect has 2 wings. Go to number 2. Hurry. The insect has long, skinny abdomen. An insect has short, broad abdomen. That's this guy. That's A. A is Apis. What is that? A is Apis mellifera. And then second we select B. 2 wings. So 2 wings go to 2. Insect has long, skinny abdomen. Long, skinny. And that is, people, Tipula simplex. Right? So B is Tipula simplex. Then, people, we select organism C. Then has more than 2 wings, can go to 3. 3, the insect has broad wings. No. Insect has thin wings, that is correct, go to 5. The insect has a broad abdomen, that is correct. Bombus. C is bombus, neither. C is bombus. And then people, D, organism D. Insect has more than 2 wings. You go to 3. 3 people, insect has broad wings, that is correct. Go to number 4. Insect has spots on the wings, no. And insects does not have spots on the wings. Catopsila. So this is catopsila. Ne? Then E people, go to number 3. Insect has broad wings. Go to number 4. Number 4, insect has spots. Then this is chateau. What is that? Chatocnema. Chatocnema. Ne? That is Chatocnema. And you know the last one is the rest. This is easy. Ne? This is easy. This is the actual use of the dichotomous key. So 6 marks for that, my dear. Going for question number 2, part C, question number 1. State the difference in habitat of Euglena and Paramecium. Euglena, marine and freshwater, Paramecium, freshwater. 2 marks for that. State 2 types of nuclei in Paramecium. 2 types of nuclei in Paramecium, meganucleus and the micronucleus. State locomotory structures found in amoeba, pseudopodia. And then what is the protestant with pocket, with flagella, Euglena. And then people, you have to compare these organisms. Complete the following table. Cell wall material, ulva, mainly cellulose, gelidium, mainly cellulose, sargassum, made up of cellulose and alginic acid. Photosynthetic pigments, ulva, chlorophyll A and B, gelidium, chlorophyll A and D, and phycobiliproteins, and then sargassum, chlorophyll A, C, and xanthophyll. How about in diatom? A, C, carotene, xanthophyll, right, people? And then people, stored food, ulva starch, gelidium, floridian starch, and then people, sargassum, laminarin. So altogether, how many marks here, people? 9 marks. 9 marks, right? For each grid, 1 mark. 9 marks for that. 9 marks, right? 9 marks. So they have asked only for 3 codes, the previous question. Yeah, okay, fine. 3 codes, you write 3, right? So, but you can write more than 3. You can write more than 3, ne? That's good, right? So we'll go for the MCQ paper now, people. MCQ paper discussion. MCQ paper discussion. MCQ paper. So find MCQ paper. Again, people, if you can come down from 25, better, ne? If you can come down from 25, better. So let's start with question number 25. Question 25. Oh. 25. Right. Question number 25. Select the correct statement regarding enzyme Rubisco. Enzyme Rubisco, correct statement, people. Rubisco is the RUBP is a substrate. RUBP is the substrate for Rubisco, that is correct. RUBP and carbon dioxide both act as substrates of Rubisco. Catalyzed to enzymatic reaction, Rubisco oxygenation reaction it carries out, and also carries out, people, carboxylation, correct. Then bind with bicarbonate. Rubisco does not bind with bicarbonate, that's a wrong one. It reacts with, people, carbon dioxide. Oxygen is a competitive inhibitor. Oxygen is a competitive inhibitor of Rubisco. Competitive substrate, it's not an inhibitor because it activates the oxygenation reaction. So Rubisco is not a competitive inhibitor, and abundant enzyme in the biosphere, that is correct, people. So we'll take A, B, and E as the answer for that question. A, B, E. right? A, B, and E. A, B, E. Moving to next question. Moving to next question. Lamai, next question. So it's not biosphere. It's stroma. It is stroma. No, biosphere is what, people? Most abundant enzyme is Rubisco. Ne? Wasn't there in the resource book? Rubisco is the most abundant. It is stroma. No, Santosh. All living organisms, neither plants and algae, uh, uh, they have this enzyme. Even in cyanobacteria, neither they do the photosynthesis. Ne? So plants are more in biosphere. Not only plants, ne? It can be algae. Right? The atoms are in the ocean. Algae are there in ocean. And then people, prokaryotic organisms are also there, even though they don't have, they have similar enzymes, right, which run this the mechanisms. So therefore, on that point of view, people, the most abundant enzyme in biosphere is Rubisco. Didn't we discuss that? It wasn't there. I remember, like, yes, it was there. At 24 people, which of the following statement wrong regarding kingdom protista? Wrong regarding kingdom protista, right? Wrong regarding kingdom protista. Natural kingdom, wrong. Natural kingdom, wrong. Artificial monophyletic group, wrong. Some are symbionts. Protista, some are symbionts. We can take as correct. Euglena is a mixotroph, but Euglena has to be underlined. Euglena is a mixotroph, correct. And chloroplast present in paramecium, wrong. So C and D. C and D. 24, C and D. Do you agree with me? C and D, 24. Question number 23. Which of the following correct regarding algae? What's your answer, people? Correct regarding algae. They are eukaryotic organisms. They are eukaryotic organisms. They are eukaryotic organisms. They are eukaryotic organisms. Algae, correct. Their sex cells are surrounded by a layer of sterile cells. Algae don't have that. Multicellular sex organs. This is a bit old question anyway. Multicellular sex organs are present, people, in plants. Neither. They are all photosynthetic organisms. Algae are all photosynthetic. We can take it as correct. They are multicellular or multicellular. Multicellular or multicellular. They are wrong. Neither. Unicellular or multicellular? I think there is an error here. Can you correct people as unicellular or multicellular organisms? And then, so that is correct. If you, after the correction, the major storage food material is starch. Major storage food material in algae is not starch. So we can't agree with that. So ACD, but there is an error. I can give you free marks on that question. Right. Moving people to next one. Question number 22. Question number 22. Which of the following correct regarding three domains? Peptidoglycan present in archaea. Peptidoglycan present in archaea. What do you say about that, people? Archaea, peptidoglycan, wrong. Neither. It was a histone associated with DNA of some bacteria. DNA of some bacteria. Histone associated. That is wrong. Neither. It was introns present. Introns present in genera of archaea. Yeah, some. And then circular chromosome present in bacteria and archaea. That is correct. And then people, methionine present as initial amino acid in bacteria. 4-mile methionine. So the answer once again, people, C and D. And then people, moving to next question 21. Next question 21. Not a difference between PS1 and PS2. Cyclic electron flow occurs in PS1 but not in PS2. That is correct. And then people, photolysis occur at PS2, but not in PS1, correct? So they are asking not a difference, ne? Cyclic electron flow occurs in PS1, not at PS2. It's not, it is a difference. So it's not the answer. Photolysis occur in PS2, but not in PS1. It is a difference. Neither. Co-complex has primary electron acceptor in PS1, but not in PS2. Co-complex has primary electron acceptors. That is not a difference. It is a similarity. It was say the two pigment molecules in center complex of PS1, but not PS2. No, that is wrong. Neither. Two pigment molecules in the center complex of PS1, but PS2. That's wrong. Neither. P680. Two pigment molecules in the center complex of PS1, but not in PS2. P680 pigment in PS2 and P700 in PS1, that makes a difference, not a feature. Now people, here, not a difference between PS1 and PS2. Co-complex has primary electron acceptor in PS1, but not in PS2. Co-complex has primary electron acceptor PS1, but not in PS2. No, co-complex has primary electron acceptors in both. So that's a wrong. Co-complex has primary electron acceptor in PS1, but not in PS2. That is, question is little unclear, ne? Question is little unclear, ne? Right? Question is little unclear, ne? Right? Mithana podi avulak thiyena. Right? Co-complex has primary electron acceptor in PS1, but not in PS2. Co-complex has primary electron acceptors in both. That's not a difference. Two pigment molecules in the center complex of PS1, not PS2. No, two pigment molecules are present in both. Not a difference. So A is a difference, B is a difference, E is a difference. Two pigment molecules in the center complex of PS1, but not PS2. Two pigment molecules are present in the center complex of both PS1 and PS2. But they ask this difference between these two. Suppose you take in the other way, a difference between PS1 and PS2. Difference between PS1 and PS2. How do you take that? We'll slightly modify the question, people, for a short time. Let's modify the question. Let's take it as, people, a difference between PS1 and PS2. Cyclic electron flow occur in PS1, but not in PS2. It is a difference. Ne? Photolysis occur in PS2, but not difference. Ne? And then co-complex has primary electron acceptor in PS1, but not in PS2. Not a difference. Ne? And then two pigment molecules in the center complex, not a difference. Ne? And then P680 pigment in PS2, but difference. Ne? So if you erase this part, then the answer is C and D. Ne? Then the answer is C and D. But anyway, there is little unclear of the question. So I'll take people C and D. Question number 20. Question number 20. Question number 20. Question number 20. Find electron microscopic structure from given list of organelles. Find electron microscopic structure from list given of organelles. Electron microscopic structure. Ribosome is electron microscopy. ER is electron microscopy. Microtubule is electron microscopy. Centriole is not. Golgi body is not. Enang A, B, C. A, B, C. A, B, C. A, B, C. Find electron microscopic structures from the given list of organelles. Enang we'll take AC as the best answer. AC as the best. AC, wait. ABC, do you know? Right? AC, let me. ACAB, ne? ACAB, AC and AB. ACAB, AD, ne? AD you can't take. Oops. AB, AC, ABD, ABD you can't take. BCE, BCE you can't take. Ne? AC, AC has to be the answer. AC has to be the answer. For 20th question, AC, A and C has to be the answer. Right? A and C has to be the answer. Right, question number 19. Right, question number 20. Right, question number 20. Right, question number 20. Take that question. Question number 20. Which of the following structure is not present in bacterial cell? Which of the following structure is not present in bacterial cell? Not present in bacterial cell. Nucleic acid present. So that's not the answer. Cell membrane not present neither. Respiratory enzymes present in bacterial cell. Ribosomes present in bacterial cells. Membrane bounded flagella not present. Fifth one is the answer. Question number 19. Which of the following is the correct way of writing the scientific name of a particular species? Dipterocarpus, this way is wrong. Ne? Dipterocarpus zillenicus. Diptera, this is wrong. Diptero, wrong. Dipterocarpus zillenicus, wrong. So we got the answer. We got the answer. Question number 18, people. Question number 18. Which of the following is not taxonomic unit? 18 ke 5, Hari. Thank you. Not a taxonomic unit. Community, ne? Good, very good. I don't have to discuss that then. Question number 17, people. Question number 17. Which of the following idea was unfamiliar to Darwin? What is your answer for that? Unfamiliar to Darwin, people. Which one of the following is unfamiliar to Darwin? There are overproduction of offsprings in nature. Darwin's idea. Man has produced many races of domesticated animals. Darwin's idea, because he saw the pigeon and the horse breeding. And then some individuals, that is why he used the word artificial selection. Eh? He knew the artificial selection. So that is why he said natural selection. Nature does the same thing. Then he knew about the artificial selection. Nature does the same thing. Right? And here people, some individuals are better adapted to the environment than the others. He knew that. Variations are produced by mutation and recombination. This is not his idea. Small heritable variations are the basis of evolution. Inherited. Ne? Fourth one is the answer. Question number 16, people. Question number 16. Which of the following name is associated with organic evolution? Organic evolution. Wallace. Second is the answer. Question number 15. Which of the following statement is incorrect? Statement is incorrect. What is your answer for that, people? Statement is incorrect. The rate of respiration of many plant tissues double if the temperature increases around 20 to 30. That is correct, because you know every enzyme activity doubles people with 10 degrees temperature increase. Every 10 degrees temperature increase is going to double the reaction rate. Every 10 degrees temperature increase doubles the reaction rate. That is accepted idea, right? But not in your syllabus now, right? Then people, glycolysis occur outside mitochondria. That is correct. Carbon dioxide release during respiration of the plant tissue supplied with oxygen. No, carbon dioxide release during respiration is not coming from carbon dioxide released during respiration of a plant tissue supplied with oxygen will contain O18. So, what's that question? Carbon dioxide released during respiration of a plant tissue supplied with oxygen 18 will contain O18. Hurry. You are giving carbon dioxide released during respiration of a plant tissue supplied. Right. You are giving oxygen with O18 isotope to carry out cellular respiration. And that oxygen, they say, comes out with carbon dioxide. Wrong. Ne? Why? When you give oxygen for cellular respiration, that forms water. So this is wrong. That is wrong. Then intermediate products of respiratory metabolism may be used in synthesis of protein. Intermediate products of respiratory metabolism may be used to synthesize protein. This is possible. Carbon dioxide is not released during glycolysis. Carbon dioxide is not released during glycolysis. Carbon dioxide is not released during glycolysis. That is correct. This is correct. The fifth one is correct. Intermediate product, this is correct. So third one is the answer. Now people, you learn amino acids can involving in cell respiration, same like that. Cell respiration intermediates can turn back into amino acids too. So protein become amino acid enter into cell respiration, the reverse can happen. That means cell respiration intermediates can transform into amino acids can transform back into proteins, right? So that is possible. Question number 13, Lamai. Question number 14, ne? Question number 14. Which of the following statement is correct regarding respiration or fermentation? Synthesis of ATP is one of the most important event in aerobic respiration. Correct. And then people what it says, which of the following statement is correct regarding respiration or fermentation? Synthesis of ATP is one of the most important event in aerobic respiration. Correct neither. And ethyl alcohol is always formed at the end product during fermentation. Ethyl alcohol is always produced. Wrong neither, because lactic acid fermentation is also there. In aerobic respiration, final electron acceptor is oxygen. Aerobic respiration, final electron acceptor is oxygen. Wrong, ne? Why? We said molecule oxygen, right, people? Molecule oxygen, wrong. Majority of the oxidation reaction in aerobic respiration is catalyzed by dehydrogenation. Correct. Dehydrogenation means, people, wait, I think majority of the oxidation reactions in aerobic respiration are catalyzed by dehydrogenation. That is correct. Why? Dehydrogenation means formation of NADH. So in most of the aerobic respiration steps, you can see either FADH2O, NADH is formed. Either FADH2O, NADH is formed. That is a process. But it says majority of the oxidation reaction of aerobic respiration catalyzed by dehydrogenation. Can dehydrogenation mean removing of hydrogen from Krebs cycle intermediates and giving that to NAD plus to make NADH. You learn dehydrogenation, neither. Removing of hydrogen is dehydrogenation. So that happens. Fourth one is correct. ATP is not synthesized during anaerobic respiration. ATP is not synthesized during anaerobic respiration. Wrong. Glycolysis involved. So we have two answers now. First and fourth. First and fourth for that. First and fourth for that question. Right, people, I want you to find the answer for question number 13. We are now limited with the time now, people. Right, anyway, we'll check this one. The number of ATP molecules already released by the aerobic breakdown of molecule of glucose in heart muscle cells. What is the number, people? Number of ATP molecules released by aerobic breakdown of one glucose molecule is 32. Ne? That is 32. And then people moving to next question. Which one of the following statement is incorrect? An accepted system of classification need not based on evolution relationship of organism. Accepted system of classification need not to be based on. They ask incorrect one. Ne? They ask incorrect one. Now, need not to be based on evolution relationship. We know people some classifications. They are artificial. The artificial classification is also there. In naming species, the name of the genus followed by specific epithet. Name genus is followed by specific epithet, correct? Virus are not named according to binomial, correct? And order represent collection of families. Order represent D, K, P, Co, F, G, S. Order and family. And order represent collection of families. That is correct, neither. Correct. A species cannot have further taxonomic subdivisions. Wrong, ne? Fifth one is the answer for the 12th question. Question number 11, people. Which of the following is correct regarding photosynthesis? Correct regarding photosynthesis under natural condition. The rate of photosynthesis in aquatic plant is limited only by the concentration of carbon dioxide in water. No, light intensity 2, ne? Wrong, ne? The light intensity 2. And then we will rate of photosynthesis affected by changing in the air temperature. Rate of photosynthesis affected by changes in the air temperature. Correct regarding the rate of photosynthesis affected by changes in the air temperature. Not air, in the leaf, neither. We'll take that air temperature. Rate of photosynthesis is affected by changes in the air temperature. Which of the following is correct regarding photosynthesis? I'm not sure. This will leave it and come back for that. Source of oxygen evolved during photosynthesis is carbon dioxide. That's water, wrong. Calvin cycle is photosynthesis limited by concentration of carbon dioxide in terrestrial plant. Calvin cycle of photosynthesis is limited by concentration of carbon dioxide. Calvin cycle limited by concentration of carbon dioxide in terrestrial plants. Limited by concentration of carbon dioxide in terrestrial plants. We'll leave that too. The rate of photosynthesis can be measured more easily and accurately by the amount of carbon dioxide by oxygen released. Now we are having second and third. Now we have second and third remain here. So check those two people. Second, rate of photosynthesis affected by changes in the air temperature. Factors affecting factors affecting photosynthesis. Factors affecting photosynthesis. We took temperature, neither. Temperature we took that as a factor, ne? Air temperature can affect. Right? Yes. So we'll take two answers then. Second and fourth lamai. Second and fourth kilogram. Question number 10. Which of the following statement is incorrect regarding enzymes? Incorrect regarding enzymes. Lamai. Incorrect regarding enzymes. Enzymes are formed by polymerization of amino acids. Enzymes are formed by polymerization of amino acid. Hari. Some enzymes contain non-protein components in their structure, correct? And then enzyme activity is affected by condition of the medium. Yeah. Enzymatic reaction do not produce heat. Wrong. Most enzymatic reactions are reversible, correct? Fourth one is the answer, people. Enzymatic reactions, people, as the seeds germinate, the seeds get heated up. Enzymatic reactions can release heat. Some reactions, we can't say that is not releasing heat. Right? So we'll go with question number 9 then. Question number 9. Any other doubts, people? Question number 9. We are in the last page now. Question number 9. Right? Question number 9. We are a little out of the time, but only 9 questions. We'll mark them quickly, people. Question number 9. What is your answer? You tell me the answer, people. I have taken answer as the third one. Third, right? The question number 8, people. My answer is first. Do you agree with me? Answer is first. Question number 7. Answer is 3. Question number 7. Answer is 3. Question number 6. Answer is 3. Question number 6. Answer is 3. Question number 5, people. My answer is BioEve test. BioEve test. Question number 4. My answer is amylase. Amylase. Question number 3. My answer is, people, MN. Question number 2. My answer is species. And question number 1. My answer is 3.5. That's all. That's all. That's all for the day, right? Did you mark them? But anyway, I'll be giving you, people, the marking scheme, right? I'll be giving you marking scheme. So then, thank you very much for your time. Thank you very much for your time, right? You can have a little nap now, and I'll see you, people, on Saturday morning 7:30. We are going to discuss Animalia lesson, right? Very good lesson, interesting lesson, right? And we'll meet you on Saturday, right? Have a nice time, right? And bye-bye.